Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we have a few announcements to make. First, we would like to thank our Dr. GPCR Ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. What are you doing on May 19th? Mark your calendars as we will be hosting another free symposium, this time on GPCR activation and signaling. Our program includes talks from Oliver Hartley, Gaspar Pandey-Sekaresh, Ilana Kotliar, Nicolas Gilles, Shivani Sajdev, Etienne Biard, Surya Shenoy, Paul Gasser, Stuart Mosley, and Kavya Krishna Kumar. We have an exciting program for you. We have also set aside 2 hours for poster presentations and networking on a platform called Hopin for the symposium. There's no limit on the number of posters we can accommodate, and everyone is welcome to present a poster at our upcoming events, including the one on May 19th. If you'd like to present a poster, go on our website and look up the event page. For more information about the event and an updated schedule, you can go to the ecosystem, and the easiest way to get to it is to use the links in the footer and look for Dr. GPCR Symposia. You can join us live by marking your calendar and becoming a Dr. GPCR Ecosystem site member, which is free. To navigate the ecosystem, please use the direct links in the footer. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I'm excited to have with me Dr. Prasenjit Saha. Hi, nice to meet you. Nice to have you on the podcast.
Nice to be here. Thank you so much. Me being here, it's a privilege here for me being here. And this is a good podcast. I have been following this podcast for a while, and I'm very happy to be here.
Thank you so much for joining me. Why don't we start at the beginning? Can you please introduce yourself?
Yeah, sure. So my name is Prasenjit Saha. I did my PhD from Indian Institute of Science, Bangalore, India. So I did my PhD in rare mitochondrial disorders, which are linked to iron sulfur cluster biogenesis. So those are the diseases which are linked to oxidative phosphorylation deficiency kind of things. So some of those mitochondrial dysfunction and other kind of mitochondrial disorder, those are the field that I worked in my PhD. So it's basically like cellular biology and molecular biology kind of work. So after I did that thing, I came here in US in 2016 to do my first postdoc here. So I joined Cleveland Clinic in 2016 in Dr. Stanley Hazen's lab. So he's working in gut microbiome and cardiovascular disease. So that was my initial interest, work after my PhD, working in cardiovascular disease and gut microbiome. So I started in 2016. I did my postdoc till 2021. In the same lab, I got promoted to research associate, which is a staff position there. So from 2021 January, I'm working as a research associate in the same lab. I'm still working in the same field, gut microbiome, gut microbial metabolites. So my thing is to identify receptors of gut microbial metabolites to find the molecular targets. So that's pretty much it. That's what I do right now.
Interesting. And where do GPCRs come into the picture?
Yeah, so when I joined the lab, so I did not it was not defined that I have to work on GPCR. So I was given a metabolite, which is gut microbial derived metabolite, which involved in cardiovascular disease, tracks with cardiovascular disease. So my boss told me you have to identify the receptor of this metabolite. It could be a GPCR, it could be ion channel, it could have been anything. So that's the first thing I started. I wanted to find the receptor of that compound. So initially, actually, I looked for GPCR because that's the most abundant kind of class of receptors and relevant in diseases, also in cardiovascular diseases. So I started screening for different GPCRs. So I used siRNA libraries to screen different GPCRs, and then I found out that my metabolite, which is called phenylacetylglutamine, which also tracks with cardiovascular disease, acts through adrenergic receptors. So that's how actually I ended up working with GPCRs. So that's my story. It's not planned before, but I just bumped into GPCR. So that's my story.
How interesting. And Let's go back a little bit in time. I've been trying to get people to go back in time and ask, did you always know you wanted to be a scientist?
Yeah, actually, I did my bachelor's in microbiology. So after that, I did my master's also in biotechnology. And then actually, I wanted to do my PhD from Indian Institute of Science, Bangalore, that I just told before, because it's like one of the like best research institute in India, and I always had dreamed to like do my PhD from there. So that's been always there, like in me, that if I do my PhD, I would do it from there. So I loved science like all my like time of my during my bachelor's, masters, and then when I got my PhD in like IISc Bangalore, so that's the time like I really got like really motivated and pumped up to do all my research there. So it was there that I wanted to be a scientist, and when I got the right platform, everything was on the right track, and it just like from then it just like took the right direction, and like now I'm here. So I'm happy that I decided to be a scientist, do my like did my PhD from the right place, and right now I'm here, and so far uh, I'm like really liking it what I'm doing, and yeah, science is like exciting. So like
It
why
is. And
not?
how did you
Yeah.
pick your postdoc? Because you could have
Uh,
gone anywhere in the world.
yeah, actually, so the reason initially, actually, I wanted to go to Europe. So the reason is like it's like quite like near from India. Like if I like take a flight, I can just like go there in like 8 hours and come back to India. But if I come to US, then it's like a like 18-hour flight. So initially that was the reason. But then actually I also wanted to work in cardiovascular disease because like my family and friends, like several other people, they had some like cardiac problems. So that was the reason that even my mom had cardiac problems. One of my close friend, he had. So that's the reason. Like I wanted to work in like cardiovascular disease, and Cleveland Clinic is it's the best place that you can like work in cardiovascular disease. Like Cleveland Clinic is like last 26, 27 years. It's the best like place where people do like research in cardiovascular disease also in like in terms of like hospital. It's the best hospital for like cardiovascular care. So and also my boss Stanley, he's like pioneer in the field. So when I got a position here in his lab, so I was like really happy, and I thought that okay, this is the right place, and this is what I wanted to do like after my PhD, and I got into the right place. So yeah, that's the reason, and why I like end up here in Cleveland, Cleveland Clinic. So yeah.
That's great. And did you? The reason I'm asking these questions is because I think the audience is on sometimes can be on the more junior side, and when you're a
Yeah,
junior, you're trying to figure out, okay, how do I pick my next
yeah,
job, and how did you, that's why I
exactly.
try to ask these questions, because learning from other people's experiences, like in your case, you mentioned that you're very much interested in cardiovascular diseases,
Yeah,
and you figured out where are the best, in your opinion, the
yeah,
best researchers, and that's why
mhm,
you ended up in the lab you are at the Cleveland Clinic, which
yeah,
is also a great kind of a sequence of events and justifying why
yeah,
you are where
exactly.
you are today.
Yeah, yeah, exactly. Like people like what they love to do and they what they want to do. Maybe that's the priority people should give. Although it's not like all the time you don't get what you want, but it's like it's worth giving it a try. So I tried, and I actually ended up here, which I wanted to like join here. So it's also like you also need to like keep all your doors open. So
Yeah.
if this doesn't work, you should have your plan B as well. But yeah, anybody should like go for the things that they want and the places that they want to go
Yeah.
and the kind of field that they want to work in that kind of field. So yeah.
Yeah. And so you mentioned working with metabolites and looking at the connection between the microbiome and cardiovascular diseases, and you stumbled upon GPCRs. When that happened, did you realize that it was a GPCR, or in your mind it was just quote unquote at first a receptor for your metabolite, and
No,
that was a good thing?
yeah, yeah. So it's like the answer would be like yes for both of them because since I know that I have to find a molecular target, it could be a GPCR. So when I know that it could be a receptor, so I had to like study all the receptors. So I studied GPCR, I studied like ion channels and like nuclear receptors. So all the receptors I studied because it could be anything. And when I like found out that it's GPCR, then I like focused into more into GPCR. So since, and it was also like our prime target was like GPCRs. Like we could have like screened like most of the receptors, GPCRs and other receptors, as I told. But then actually I was like, I would say I was fortunate that I wanted to like screen for GPCRs and I got GPCR that it's going through a GPCR. So I was fortunate and like it saved me a little bit of time initially, and then actually the rest is like after that I only like working on like GPCRs like since then. Yeah.
So tell us a little bit more about this metabolite and how it
Yeah.
acts
Exactly.
on the
Yeah.
beta-adrenergic receptor, which
Yeah. Yeah.
it's the GPCR, or the
Yeah.
grandfather of all GPCRs when it comes to
Exactly.
getting to know it.
Exactly. So uh, so initially like this compound it's called like phenylacetylglutamine. Like in short, we call it as PAG. So PAG is it's a gut microbial derived metabolite. So and when it's like in the circulation, it's like it tracks with cardiovascular disease. So we have in our lab we have like a gene bank sample of like 4,000 patients, and we since we have access to those patient samples, so we look for like molecular signatures in their blood. So we look screen for like all 4,000 samples, and we try to see what are the metabolites that are like elevated in those patients. So PAG is one of them. So we saw that PAG tracks with cardiovascular disease, especially in like quartile 4. The level is like really high. So obviously we wanted to know the molecular reason that why it's like tracking with cardiovascular disease. And since actually then we screened for different GPCRs and we found out that it's going through adrenergic receptors, and more specifically beta 2, alpha 2A, and alpha 2B adrenergic receptors. So there are like total 9 adrenergic receptors there. And we screened for all 9 of them. So initially we did with siRNAs, and we found out that the best targets of out of all these 9s are beta 2 is the first one, and then alpha 2A, and then alpha 2B. So and after that, like we tried to characterize the interaction between the receptor and the metabolite. So we did like various kind of like cell signaling assays, secondary messenger assays, binding assays, and all those different things that people would do in like GPCR field. So we did like most of them, and we figured out that it's acting through adrenergic receptors. So that was the like first part of our research. So we published that part in like 2020 in Cell, which tells that it's acting through adrenergic receptors, and more specifically like alpha 2, alpha 2B, and beta 2 adrenergic receptors. So that's pretty much it. Like our initial finding with the metabolite and its receptor.
And is this metabolite, so you mentioned that it's increased in patients who are at high risk for cardiovascular diseases,
Mhm.
but I'm just trying to understand, when you're not at risk, is this metabolite still present in
Yes,
patients, or
it's
is
it,
it
it
at a lower dose?
it is at a lower dose actually. It's like really lower dose. So basically that metabolite it comes from any protein that we eat. So it comes to like phenylalanine. So phenylalanine is there like any protein that we eat. So mostly it's like in protein diet, or in like any of the meat that we are eating, or any kind of meat that is like enriched with phenylalanine. So those are the thing that contributes to like elevated level of PAG. Even if like some of those like soft drinks like Diet Coke and all, so they do have like extra supplement of phenylalanine. So
Oh.
those are the things that if people drink or eat that is having like high level of phenylalanine, that will like eventually give rise to like more amount of phenylacetylglutamine. And also like I'm pretty sure like the basal amount would be like really in nanomolar or something in that sense. But in patients like who are like cardiovascular disease patients, or they generally we like divide in like 4 quartiles, quartile 1 to quartile 4. So quartile 1 would be having the lesser amount of PAG, and quartile 4 would be the higher amount of PAG. So in quartile 4, like it's somewhere around like it can go to like 10 micromolar to like few hundred micromolar. And also like people with like chronic kidney disease, so they're not able to like excrete those metabolites, so the levels will like gone high, like really high. So in those patients that it might go up to like millimolar level. So the more the amount and more problematic it would be actually.
And how does it act differently on the three different adrenergic receptors. And it makes me wonder, is this an allosteric modulator?
Yeah, exactly. So, that's the second part of my study. So first, actually, we thought that, okay, it is acting through a beta-adrenergic receptor, but then what exactly it is doing? So initially, we thought that it might be a partial agonist because we see that by itself, it increases cyclic AMP level. So we thought that, okay, and the amount is like very less. It just like follows the graph of a partial agonist. But then we did not know that we tried to like go like deep into the situation that what exactly it's doing. But then actually we did not have any clue that if it is acting as a partial agonist, then how it's like contributing to cardiovascular disease. So we thought that like in our department there is a also a scientist who work in GPCR. So he actually told us that why you should like think it in a different way, that you might think that it might also work as an allosteric modulator. That it's something that really people like working. This is like really a new field, and people are really working in this field, and this is something new, and that you might have to like think it or see it in that way. So then actually I use some agonist, and in presence of agonist, we do see that PAG is like reducing the function of like known agonists like isopropanol or norepinephrine. So then we thought that okay, it is acting as a negative allosteric modulator. So that's the second part of our study. We haven't like still published it yet. So those things are like still going on, and like we kind of like figured out what's the exact mechanism and where it might bind to the beta 2 adrenergic receptors. So far, everybody knows that like people are trying to like develop allosteric modulators, and beta 2 adrenergic receptors like so far like uh, like Dr. Lefkois and Brian Kowalka, they found that there are like three allosteric sites present in that receptors. So we initially like we screened for those three allosteric sites, and also we did some like in silico docking to find out what could be the pockets other than the orthosteric site where PAC can bind. So we actually found out where exactly PAC can bind. So those are studies still we haven't like published it yet, but yeah, we have like clear idea that what's the mechanism and how it's like doing those things. So yeah, and even like in terms of biological response, or if you want to see like any broader response. So we also like, since we work at Cleveland Clinic, and we do have like patient heart samples, like we get patient heart tissues. So we actually try to like see the contraction with norepinephrine, those contraction of the heart muscles. And then when we put PAG, we see that it nicely reduces the contraction. So we thought that, okay, even like its negative allosteric modulated behavior, it's translating to heart contraction. So that's like quite fascinating, and like so far people just like developed all those like allosteric modulators which are like synthetic, and there is something like that endogenously like produced in the body, and which is acting as like allosteric modulator, which is like really fascinating for us and for me as well, like me as a scientist. And because there was like a roadblock at one point of time, we did not know that what's going on, and then we figured out that okay, this is what exactly is happening. So yeah, so far this is how it's acting. It's acting as a like allosteric modulator.
How cool is that?
Yeah.
Any favorite GPCR out of the three that you've identified?
Yeah, actually, I like the whole class of this adrenergic receptors. There are 9 adrenergic receptors, and it's so diverse. Like in 9 of them, you will get like those are like coupled to like G alpha those are coupled to G alpha I, G are coupled to G alpha Q. So all three are like covered in that same thing. And the beautiful part is that one part of the alpha receptors, they actually involved in like vasoconstriction. So it's like um, increases the blood pressure, but then betas, they relaxes the blood vessels. So they do like quite opposite work. And personally, for me, definitely it's beta 2 is my favorite because it's like been studied well. That's the first crystal structure people like in 2007. That's the first GPCR that has been like the structure has been solved. So, and also since like it has like that like clinical implications, and several are there on beta blockers. So those beta blockers are like acting via beta 1 or beta 2 adrenergic receptors. So for me, betas are like my personal favorite to like study. And since like also the allosteric studies are also going on in beta 2, like other than as compared to all the other adrenergic receptors. For me, the personally beta 2 is my like personal favorite
I
for
love
sure.
it. And you're right. I mean,
Yeah.
it's everybody has the beta 2 at some point, worked on a beta 2, even if it's just as a control to set up the
Yeah.
other assays. And it's a really nice model
And
system.
exactly. And also, I like one cool fact of beta 2 adrenergic receptor is that, like, when it's present in heart, like, if there is a beta agonist, it like contracts the heart. But if it is like in somewhere else, like for in terms of like lungs, smooth muscle, so when there is a beta agonist, it's like relaxes the muscle. So the same receptor and same agonist, but it does like entirely opposite function. So that's like pretty one of the cool thing that like going on with beta 2 adrenergic receptors, which like makes me like more fascinated towards beta 2 adrenergic receptors.
And location matters. And I don't think
Yeah, exactly.
sometimes we don't realize how much it matters. And sometimes we don't have the right tools to look at that. So
Yeah, exactly.
amazing, amazing. And I want to go back to the findings that you had. I have to read the paper because now it's like it's super super interesting to me. Any differences of the metabolite on the function of the three different GPCRs? So the beta 2, the alpha 2, and then alpha
Yeah,
2B.
yeah. Alpha 2A and alpha 2B. So also it was like a little bit fascinating for me because beta 2 is like G alpha S subunit linked, and alpha 2A and 2B they are like G alpha I. So one would like increase cyclic AMP and one would like decrease cyclic AMP. So that was like entirely opposite. So we thought that okay, what's the net result? So as a net result, we do see there is like increased cyclic AMP. So we thought that okay, maybe beta 2 would be the prime target for the metabolite. So that's the thing actually. So we haven't like there are like several things that has to be done in this field, or at least like what I'm doing right now. So we know like we check like cardiac function. So it makes sense like when we check cardiac function. So where betas are like really beta 2 is like a prime thing, beta 1 and beta 2 is a prime receptor for there. So we do see that they are involved in heart muscle contraction, and PAG is somehow reducing its function. So there is like lesser heart contraction, and that leads to like heart failure and cardiovascular disease. But when it comes to like alpha 2 and alpha 2b, they're linked to blood pressure. So it's present in the blood vessels. So if it will activate that receptor, so there will be like more blood pressure. And if it is not, if it's like acting as a negative allosteric modulator, same in that case, we would see there is a lowered blood pressure. So that actually haven't studied it well. Like we did not check the allosteric modulator angle in alpha 2 and alpha 2b as well. But so since we have like those clinical data, so we see that like PAG tracks with like lowered blood pressure as well. So maybe it is also like acting on those alpha 2 and alpha 2b receptors, and as like a negative kind of effect it's giving there. But those things are like really we are like really interested to look at those things and haven't done it yet. And as I'm saying, there are like a lot of things that can still we can do and try to like figure it out that what exactly it's doing in like different receptors.
From a patient standpoint, since the PAG increases in cardiovascular patients suffering with of cardiovascular diseases, from a patient standpoint, what would we want? Would we want to decrease the concentration of this metabolite to help them manage their disease,
Yeah,
or what does that look like?
yeah, definitely. So ideally, like we should decrease the level of the metabolite. So we also there is a part of in our lab there is a group of people who works in like how we can like reduce the amount of the metabolite. So since it's like a gut microbial derived metabolite, so they target those enzymes. So there are a bunch of like different bugs in the gut. They
Mhm.
produce those metabolite and they have like designated enzymes. So basically what we do, we try to get like substrate analogs of those enzymes. So and then we like try to find out we just have our like mice models, and then we just like treat those like different molecules that we try to develop that can like target gut microbial gut microbes and their enzymes, and then we see that if the level of the metabolites are less in the blood or not. And at the same time we also look for like different cardiovascular disease models like atherosclerosis or clot formation or thrombus formation. So we do have like all those models been like set up in the lab, and we try to target different gut microbe and their enzymes, and then we as a readout we try to see what's the plasma level of this metabolite and how it's affecting the other cardiovascular disease phenotype. So that's one way to like look at it. And the best thing would be like to restrict the kind of diet that is having like more of
Yeah.
phenylalanine. So that's the easiest way people can like do at this point of time.
Which is actually was going to be my next question. Can you with
Yeah.
diet influence?
Yeah,
Um,
diet, yeah, diet, it's such a like a nice thing actually that like if we have to like make our lifestyle better and like to feel like physically better, like diet has like a huge impact. I don't know like if you there is like very recently there's a paper that has been published in from our lab. It says that like erythritol that is also artificial sweetener that is also like
mhm,
tracks with cardiovascular disease and like also like I since I told that I was trying to look for receptors for gut microbial metabolite. Also, I tried to like see for like receptor for like different artificial sweetness as well, and some like secondary bile acids. So that is also one thing. Like erythritol, it just like published like few days back in Nature Medicine. So it says that um, it tracks with cardiovascular disease and the increased level of blood level of erythritol that like leads to thrombus formation. So it like aggregates platelets. So if there is a clot formation that increases the risk of cardiovascular disease, stroke, and death. So that's the thing. Like whatever we eat, that's like really important.
yeah,
Like doesn't matter what are the metabolites that are producing, or it doesn't matter if it is like a
yeah,
artificial sweetener or anything of that
yeah,
kind of sort of those things. So it's just like pretty fascinating for me as well. Like me being scientist to find the molecular targets of those things. It could be a gut microbial metabolite that tracks with like adverse outcomes, or it could be um, artificial sweetener, which like most of them, like now people are like showing most of them, they have some like negative health impacts. So those pretty fascinating, and like to find out receptor for those things, like it's very challenging for me as well. At the same time, it's like really exciting. Like once you get that your eureka moment, yeah, this is the receptor, it might function, and then it's just like you're so happy that whatever you're doing, it's just like getting in the right direction.
wow. And you mentioned erythritol. If I remember correctly, it's extracted out of plants. It's not that,
Yeah,
but then the question is, how much erythritol do you need to consume in order to
yeah.
actually get to that um
Yeah. So
the state?
yeah, it's actually the thing is that erythritol was why it was popular. It is like naturally present, like some fruits and vegetable, and also it's like endogenously produced in the body. So that's why it was like popular. So it is there in the body. But the thing is that the supplements or like the keto diet or the drinks that
Yeah,
we have, those having like at least like thousand-fold higher amount of the basal level that we have. So that's the reason it's like it might be a problematic when you are like taking a keto ice cream. So most of them
yeah,
they will be like putting erythritol in that. So
yeah,
you'd be having that you're like taking less calories, but at the same time you're taking erythritol.
yeah,
So that can like really contribute to like adverse outcomes, like negative
yeah.
health impacts. Yeah.
Yeah. Well, we've had so I did my postdoc at Rockefeller, and one of the scientists in the lab used to drink a lot of Pepsi, I think, or a Coke or something, some kind of a soda. And at some point he did this experiment on himself where he was measuring his blood glucose level, and everything he was drinking was diet, but
Hmm.
he did see the increase in blood glucose after consuming the soft drinks.
Yeah.
And these were all made with aspartame, I think, at the time.
Yeah. So that's like that's present in Diet Coke and most of those like yeah soft drinks. Yeah,
Yeah,
actually, there are like a couple of studies. Actually, they tell that even like aspartame are like they tracks it like cardiovascular disease.
yeah,
So I would say like maybe you should like take even sometime I also like have those artificial sweetness. The key would be like to have them like in lesser amount. I would say,
yeah,
or just like stick to like regular sugar. I guess.
yeah. Fruits,
Yeah.
fruits, stick to fruits
Yeah.
and
Fruits. Yeah.
honey. Yeah, because
Yeah.
with fruits you have the fiber as well, so it kind of I don't want to say it balances it out, but having the fiber also helps with the bump in the blood
Yeah.
glucose
Yeah.
level.
Yeah. Yeah.
So we went from GPCRs to
Yeah.
artificial
It's like sidetracked a
sweeteners.
little bit.
Um, um, within your research in the lab, if are there any tools that you wish you had? Some kind of an assay setup, some kind of a compound tool, or anything. If you had a magic wand and you said, "Oh, I wish I had this," what would that be?
Like the thing is that if I have that or not, actually I already have one. like I just like really wanted to talk about it. So like initially, like when I had to like screen for different receptors, so like my in my lab we actually came up with like level 3 assay technology. So it's called like dynamic mass redistribution. So it's kind of little bit like, I wouldn't say like it's really popular in the GPCR field, but it's such a like a novel and like cutting edge level 3 assay technology that can like detect receptor. If you have a like a compound and you have a cell, so if that compound is acting through a receptor or not, it will tell you. So that's the beauty of that thing. So you don't need any kind of like radioligand thing. You don't need any level for that. It's like level 3 assay technology. So initially my initial screening platform was like dynamic mass redistribution. So in short we call it like DMR. So when I was doing DMR, so the basic thing is that, so when you have a cell and it does have like all the receptors on the surface, or it might be having any internal receptors. So when you are putting a compound, if it is a binding to a receptors, there is like whole cellular redistribution. So that's a dynamic mass redistribution of the cell, that the whole thing changes in the cell. So if there is a change in the cell, so it will be detected by that machine. So ideally, what happens if a compound binds to a receptor in a cell? So when these like cellular constituents inside the cell, they move, they either it will move down or it will move up. So based on that movement at the bottom, there is a sensor and it detects that movement. So if some compound binds to receptor, you just like measure that movement, measure that dynamic mass redistribution. But if it is not, there is no signal. So that's my initial screening platform. So I knew that my PAG is like acting through a receptor because it's giving a DMR signal. And then I used like different siRNAs. We had a GPCR siRNA library, and then we treat those cells with the individual siRNAs, and then we looked for that readout if we are seeing any DMR response or not. So then that's the initial screening, and it's such a beautiful like screening platform. You could like have 96 well, 384 well plate, and then you can just like screen so many different cell lines. And at the opposite way, you can like screen so many siRNAs or so many different agonists or antagonists. So in both way, you can either look for different metabolites or different receptors. So both way you can like work with that thing. So that's one thing I really loved doing experiment on those machines, DMR machines. And also since that was my initial screening platform, and then I like ended up with GPCR. After that, like started doing all those like secondary messenger acids like cyclic AMP, calcium, and then also beta restin. And also one fascinating thing, since you like talked about if somebody wants to have that tool or not, that Presto Tango beta restin recruitment. It's been developed by Brian Roth. I think he also was there in that one of the podcasts I have seen.
Yeah.
So yeah, so they actually developed a beautiful like cell line which is called HTLA. So it's called like Presto Tango. So there you need to like have two genes to have that tango. So when like if you're adding a compound, so there is a beta arrestin gene, and there is, if it is activating, the beta arrestin goes, it binds to the internal side of the receptor, and it chops off the C-terminal part, which is having a tetracycline trans activator, and it goes into the nucleus, and then there is like expression of luciferase. So that's pretty cool thing, I would say. Like after that, like that is like one of my best screening platform for my different like gut microbial metabolites. And it's since like most of them are like standardized, and they have in that paper they have shown the different like positive control and negative control for all those things. So that's pretty cool platform, I would say. It's like really nice platform for me. Like I love working on those Presto Tango GPCR library. And since we have that library, and since we have a library of different metabolites, so like I have like all those things, library of receptors and library of metabolites. So there are like plenty of things that I can do actually.
It's nice. It's kind of a mix and match and then see
Yeah,
what lights up.
exactly. Yeah,
That's
exactly.
amazing. That's
Yeah.
amazing. Obviously the answer to the next question is yes, but if you could maybe expand on it. I always ask, do you think GPCRs are still good drug targets?
Yes. I'm like, this is so fascinating for me. Like me being, I'm like so passionate about like GPCR drug discovery, and especially GPCRs. So there are like almost like 800 GPCR present, and almost like 400 of them are druggable. And I think if I'm not wrong, maybe like 150 to 165 have been like targeted. And And we all know like that like one third of the all the FDA approved drug, those are like they're targeting GPCR. So since there are like 800 of them and 400 of them like that can be like druggable and that can be targeted, they're like still we have like 400 left. So and there are like so many orphan receptors that hasn't been like characterized yet, so we don't know what's their function and how we can like target them. So still there are like a lot of window to like do work. And since it has I would say GPCRs are like so abundant and so diverse. With the abundance you have like a your area is like huge. You can like go for like all different directions and like target all different GPCRs. And at the same time, like since it's like the functionally so diverse, it covers all different disease aspects, like you name it, like from cardiovascular disease, cancer, diabetes, obesity, everything is like linked. It could be like linked to GPCR. So those are the things that definitely like with GPCRs, like targeting different GPCR, those can be like something people can address those diseases. And like so far has been it's been done like tremendous amount of work has been done with like GPCRs. And like it has like it helped like patients like immensely, I would say. And also there are like other things like which can people still can look at it and can be definitely it can be like really helpful to people and like patients. And there are several things that we don't know at this point of time. I'm pretty sure that those things are like I would say definitely GPCR like still my present like favorite like class of GPCR, the class of receptor that uh, can be used as like a drug discovery platform. Definitely, yeah.
No, great, great. I think I don't think anyone answered no to my question. That would
Yeah,
have
no,
been
actually,
an interesting
yeah, you know,
conversation.
like some people like it's like people who work in their field, they just like really passionate about that thing. But people also like now, you know, with the gene therapy and like cell therapy, people like really passionate about those things. So those are also like really something that's like curing people, like things that like people might not have like imagined like few years back, that those are also the thing that can be like used to treat like difficult diseases. And the things that we are those are like not like accessible by GPCR. So some immunological disorders, like not everything can be covered with GPCR. So definitely, yeah, there are some like pros and cons, but still like for me as like being a GPCR biologist, so I'm pretty sure like there a lot can be done in the GPCR. So there are like several things that has to be answered.
There's plenty
Yeah,
of work to go around
yeah,
for everyone, for
yeah.
sure. All right, top three aha moments that shaped your trajectory.
I would say actually my first would be like my identification of receptor of my metabolite. So that's really and like it's a great moment in my life. You know, actually I always like people always tell about their like success stories. So when I joined here, so I my metabolite was different. So I was working on a metabolite. It's called TMAO, trimethylamine oxide. So which is also like a popular gut microbial metabolite that tracks with cardiovascular disease. So initially, I have been given that compound, like identify the receptor of that compound. So I initially actually have 1 year I worked in that thing, but then actually I did not get anything. But then actually as a plan B, I had like other metabolites also that I was working at. So when I like find the receptor of the metabolite that I were working like after two and I would say 2.5 years, it was like really like exciting moment for me. Like it's a really aha moment for me that okay, I got this thing. And I also I would say maybe like for anybody, I would say that maybe the first paper would be like really good aha moment for somebody. Even in my PhD, when I got my first JBC paper, it was like really exciting for me that time. So that is also I would count like one of my favorite things actually, like being like so happy about like doing science. So yeah,
Yeah, it's really motivate. You get very
yeah,
motivated when you get that
yeah, yeah, exactly,
email that says congratulations, your paper
yeah,
has finally been
yeah,
accepted after
exactly,
I don't know how many
exactly,
rewrites and versions and additional experiments. So it's a big deal.
yeah, that's true.
Any advice for young scientists?
Um, I think like for all the scientists, like if somebody wants to be scientist, I think whatever the field they want to work, I think they should have like a solid background in that field. Like you just need to like
Yeah.
know a lot of literature, what people have already have done, and what the things are. So if you have like a solid background and solid base with your topic of research, you can always come up with like new ideas. And since I'm talking about like new ideas and like development in your goals and project, also you need to be like updated with the things that are going on in the field.
Yeah.
So all the previous literature and what's going on in the field, all the new things that things are coming up in your field. So that's the second thing I would say. Also, I think that the collaboration is like a good thing. Like you can, if you just like get a chance, like talk to different people in your field, like they might be having different ideas and different aspects, so they can like definitely tell you about collaboration is also something that new that the people that who wants to be a scientist or who are new in the field, they can do something about it. Yeah, I think that's pretty much it. And also, like, yeah, you have to like increase your network. The same thing, like actually with the network that I built with people, like same, I just like talked to you on LinkedIn, and then I actually ended up here. So that is also one important thing. Like the more network you make, I think that's like that helps a lot actually. You being a scientist.
Yeah,
Yeah.
let's stop at the networking aspect, because I don't like the word networking because it's such a cryptic word. So if you were to give an example as to how you would, if a junior scientist comes to you and says, "Oh, I really want to network, what do I do?" In your opinion, what would you tell them?
you know, first of all, like me personally, actually, like I am like kind of like friends with all my batchmates and all my seniors, like from my grad school, from my postdoc. So I try to be in touch with all of them, and also I try to be in touch with all the people who are like big in GPCR field. And uh, for example, like there are like two people, like both of them, they're like one of them, like he's a student of lefco, is like who is in our department. His name is Dr. Prasad. So actually I go to him, like I if I have any problems with GPCR, if I have to like gain new knowledges. So yeah, I go to him, and I just like it just like increases my knowledge. So my idea of networking is just that, like, try to be in touch with all the people that you have worked before you're working, and that you think of that you might work with them. So you try to like be in touch with them, and also the people who are like popular in your field. Try to make some like role models. Like this is the guy I should like follow their work. So yeah, that's how maybe a networking would be like a good thing.
Yeah, I mean, you gave me you while I was listening to you talking, you gave me the idea. I think it's all about being human, and just be nice, talk to people, ask them if you can help them with anything, and just strike up conversations. I think we all love GPCRs, and that's a
Yeah,
great conversation starter. And
yeah.
just be nice, just be human.
Yeah, that's true. It's at the end of the day, it's that everybody is human. Like be nice to somebody is
Yeah,
the best thing one can do. Like,
yeah.
even I also feel the same way. Yeah,
Amazing, amazing. All right,
yeah.
I have one last question for you. What is next after Cleveland Clinic? Where do you see yourself in the next 5 years? And this is
Yeah,
not a
that's
job
like
interview. This is a job interview
I know
question.
it's yeah, it's a job interview question. You know, like see like I whether I would like stay in academia or if I would go in industry, I would like stick to like GPCR field, like any place where people working with GPCR. So here actually, if I like want to stay in academia, I have to like write grants and everything. It's like it's a big deal actually. So even actually I got some like AHA grants before, but I recently applied for a few grants. I did not get it, but it could be like a little bit frustrating if you want to stay in academia, be a PI, and like guide students. It's like a big deal. Like definitely it's a big deal. So at this point of time, I like keep all my options open. I also like would look for like industry jobs, but may not be like right at this point of time because I do have this project going on. And it's kind of almost kind of like done. I am doing some of the in Bible works that really needed to be done for this project. So and for me, I definitely, whether it doesn't matter what's the setting, maybe in next 5 years I want to lead a team who is like passionate about GPCR, working on GPCR drug discovery, and like try to like solve people problem, like people's disease that might be like linked to GPCRs. And even if it is like a little bit confusion in the field, I think that would be like enough for me to like be happy about the things that I'm doing. Like there are like several things. Like you just like develop one small agonist or one small agonist for one specific receptor. It could like really like change a whole perspective of a different like a disease model. So that's my goal. Maybe in the next 5 years, I would try to like develop different agonist, antagonist, allosteric modulators, especially like allosteric modulators, or maybe some biased agonist. Biased agonist is like pretty popular right now, because it could be like really specific, and then you might not be having like any side effect. Same for allosteric modulator, it could be like really specific. It's just like out of all different class of receptors, it might target to a specific one. So definitely in the field of GPCR, working in the field of GPCR allosteric modulators, biased agonists. So that's how I see myself in like next 5 years, like leading a team of passionate uh, people working on GPCR, trying to solve a disease, maybe a cardiovascular disease, or maybe it could be like any other disease.
Okay, so you're not stuck on
No,
cardiovascular
no,
for now.
it's not. But that's the fun thing. Like maybe I'm working on cardiovascular disease, or maybe I'm working on receptor. But the thing that I'm doing, like trying to develop an agonist, it could be the process is the same, but
Yeah.
just looking into like different diseases.
Yeah, no, agreed. And, research applied on a GPCR that is implicated in disease one can very much be applied to disease two. We just have to
Yeah,
figure out the connection there. And
yeah,
sometimes, it's very popular to repurpose drugs and
yeah,
compounds as well.
yeah,
And it,
yeah,
it can, short circuit a lot of research because, toxicity wise these have been evaluated. We have a pretty good
yeah,
understanding as to what they do to the receptor. And it could be a really cool
yeah,
set of projects. Well, I cannot wait to see what's coming for you next.
yeah.
Thank you so much for joining me on the podcast. I
Th-
really, really enjoyed our conversation.
Yeah, thank you so much. Like it was so nice. Like I was thinking that initially I was thinking, okay, this is like I don't know what I'm like talking about here, but it's like it flowed like really nice, and it's just like general talking, and I always like excited to like talk about like my science and
Of course.
specifically like GPCRs. And yeah, it was really nice to be here and like talk to you about like my research.
Thank you so much for joining me.
Yeah.
Don't go anywhere. I'm just going to stop recording. And
Okay.
we're going to let people wonder what are we talking about after we stop recording. All right.
Yeah.
Thanks again.
Thank you so much.
Thank you for joining us for this Dr. GPCR podcast episode. We would like to take a moment and thank our guests, as well as our Dr. GPCR team members, Attila, Ines, Montserrat, Ivana, Andreina, Balint, and Julia. A huge thank you to our ecosystem partners for their support: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem. Join us today. Please also don't forget to subscribe to the Dr. GPCR monthly newsletter. Find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial on our website. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. You can always email us with any questions or suggestions at hello@drgpcr.com. And until next time, stay safe.